Now that we've covered cell structure and function, we can turn our attention to finer details of cells, membrane structure and function, and cellular energetics. Here we'll cover cell membranes. The next video is about cellular energetics and enzymes. When we talk about membranes, we think first about the plasma membrane that surrounds the cell. That's as it should be. Most of what we're going to talk about applies to the plasma membrane, and the plasma membrane is the only membrane common to all cells. It's important to keep in mind, however, that eukaryotic cells have many internal membranes. In fact, if you add up all the internal membranes of a eukaryotic cell, they're much bigger than the plasma membrane. That said, let's get back to the plasma membrane and its functions. The first function we should consider is separating the cell from its environment. You can't really imagine life without some distinction between the life form and the environment. That's the job of the plasma membrane. As such, the plasma membrane also regulates the movement of molecules into and out of the cell. That's the function that we'll talk about most, but the plasma membrane also has other functions. It mediates interactions between cells so that they can work together. It also mediates interactions between cells and pathogens, like bacteria and viruses. Coronavirus, for example, gains entry to the cell by first binding at the plasma membrane. Receptors in the plasma membrane respond to hormonal signals secreted by other cells. Finally, in eukaryotic cells, membranes also enclose organelles and allow them to carry out specialized functions. But we'll only touch on these other aspects of membrane function. Before we discuss the structure and function of biological membranes, we must first learn two physical processes concerning the movement of molecules: diffusion and osmosis. This lesson comes at a good time because it happens about the same time as our lab on diffusion and osmosis. Diffusion, simply put, is the movement of molecules from an area of higher concentration to an area of lower concentration. The illustration on the left imagines a little bit of dye dropped into a beaker of water. Initially, the dye is concentrated in one part of the liquid, but over time, the dye spreads out until it is evenly distributed throughout the liquid. That's diffusion. Why do molecules spread out? The answer lies in Brownian motion, which is the tendency of all molecules to be in constant random motion. In other words, molecules have kinetic energy. Temperature is a measure of kinetic energy. The higher the temperature, the greater the kinetic energy, and the greater the rate of diffusion. If molecules are constantly moving in random directions, they will eventually be evenly spread out. This is a state of equilibrium. Importantly, at equilibrium, molecules are still moving, but because the movement is in random directions, there is no net movement. There is only net movement if there is a concentration gradient. This is illustrated well in the diagram to the right, which shows diffusion across a biological membrane. The colored molecules are initially concentrated on one side of a selectively permeable membrane. We call biological membranes selectively permeable because they allow some molecules to pass through, but not others. We'll talk about the nature of selective permeability in a little bit. Over time, molecules move from higher concentration to lower concentration, top to bottom in our illustration, until eventually there is an equal concentration on both sides of the membrane. Go ahead and count them, you'll see they're the same. Note that the molecules are still in motion, but there is no net movement because the motion in one direction is compensated by motion in the opposite direction. This, in fact, is the definition of equilibrium: forward and reverse processes happening at the same rate. One last thing to say about diffusion is that different molecules diffuse independently of one another. If there were another kind of molecule diffusing across the membrane in either direction, it would have no effect on the molecule you're seeing here. Now let's turn our attention to a special case of diffusion, the movement of water molecules, which we call osmosis. Look at the beaker on the left and you'll see a situation like we just saw. A solution with different concentrations of dissolved molecules, or solutes, on opposite sides of a selectively permeable membrane. In this case, however, the membrane is permeable to water but not to the solutes, closer to what cells are like, as we'll see. Water behaves like any other molecule and moves from an area of higher concentration to an area of lower concentration. With water, however, we don't describe its concentration. That wouldn't make sense. Water is the solvent, the thing that other molecules are dissolved in, so it doesn't really have a concentration. Rather, we talk about the concentration of the solutes dissolved in it, or tonicity. Tonicity of a solution is the concentration of all solutes dissolved in it. Higher tonicity means higher solute concentration, higher tonicity means higher solute concentration and lower water concentration. Lower tonicity means lower solute concentration and higher water concentration. Water therefore moves from an area of lower tonicity to an area of higher tonicity. Let's go back to the beakers. Osmosis continues until solute concentration, tonicity, is the same on both sides of the membrane. In other words, it reaches an equilibrium. The left side of the beaker has less solute, but also less water, so concentration is the same on both sides of the membrane. Just to be a bit more formal about it, here's a definition. Osmosis is the movement of water from an area of lower tonicity to an area of higher tonicity across a selectively permeable membrane. That's the same as saying that tonicity is the ability to attract water, and that's how some people define tonicity, but I think this is a more useful definition for tonicity. Tonicity is the total amount of solute dissolved in solvent. It also goes by names like ionic strength or molarity. Why does water move to an area of higher tonicity? I'll try and explain that on the next slide, but if you don't get it, don't worry about it. Know these definitions and you'll be fine. This is why water moves from lower tonicity to higher tonicity. What you see here is a solute represented by the hexagons. Glucose, for example, is a solute, and it's the inspiration for the hexagons. The solute concentration is higher on the left than on the right, and they are separated by a selectively permeable membrane. The little H2Os represent water molecules. Note that some of the water molecules associate with the solute. That's what it means for something to dissolve in water. There's an interaction between it and the water molecules. We learned that in a previous lesson. Not all the water molecules associate with solute. We call that free water. The red molecules represent free water, and that explains osmosis. By virtue of the fact that there is less solute, the right side has higher free water concentration, and therefore water migrates, or diffuses, or osmoses, right to left. Now let's apply osmosis to real live cells. Knowing what osmosis and tonicity are, we can now define three new terms. Hypertonic means that a solution has higher tonicity than a cell. Isotonic means that a solution has the same tonicity as the cell. Hypotonic means that a solution has a lower tonicity than the cell. I compared everything to a cell, but the larger point is that these are comparative terms. When we call a solution hypertonic, it means that it has a higher tonicity than some other solution. Now let's see what happens to cells in these various solutions. Let's start with an animal cell, which is depicted on the top row, in isotonic solution, here in the middle. As an example of an animal cell, we use a red blood cell, also called an erythrocyte. They are the cells that carry oxygen in the blood, and they are very permeable to water. Later on, we'll see why they are so permeable to water. Remember that molecules are in constant random motion, so water is always entering and leaving the cell. In isotonic solution, the rates of movement in and out are the same because there's no net osmosis. The cell is perfectly happy and it retains its shape. In hypertonic solution, on the left, tonicity is lower inside the cell than outside, so water moves out of the cell. As water moves out of the cell, the tonicity inside the cell increases until the tonicities inside the cell and outside the cell are the same. In other words, the cell has achieved osmotic balance or osmotic equilibrium. In fact, you can think of osmosis as water's attempt to reach osmotic equilibrium. So in hypertonic solution, water moves out and the cell shrinks until equilibrium is reached, and then net movement of water stops. The same cell in hypotonic solution behaves differently. Look at the upper left. Tonicity is lower outside the cell than inside, so there is net movement of water into the cell. Water will continue to move into the cell in an effort to reach equilibrium. Because the volume of the solution is much higher than the volume of a cell, it can never reach equilibrium, and the cell continues to take on water until it bursts. In terms of movement of water, plant cells are the same as animal cells. What's different is the consequences for the cell, and that's because plant cells have cell walls. In hypotonic solution, lower right, water moves into the cells and the cell swells, but it doesn't burst because the cell is contained in a cell wall. Since the cell is swollen, the cell itself is exerting pressure against the cell wall. That's called turgor pressure, and the cell is said to be turgid. This is the natural state of plants. They like to be in fresh water, and turgor pressure helps keep plants upright. Plants in isotonic solution, in the middle, are at osmotic equilibrium. Movement water into and out of the cell happens at the same rate. Importantly, the cell is not exerting pressure against the cell wall, and the cell is said to be flaccid. If this happens with a plant in your house, you would say it's wilted, and you would water it, and hopefully it would return to an upright shape. Finally, the plant cell in hypertonic solution would shrink, just like an animal cell. But once again, the situation is changed by the presence of a cell wall. The cell shrinks, but it is attached to a cell wall, so there is a limit to how much it shrinks. Such a cell is said to be plasmolyzed. Plants, therefore, are more flexible in terms of how much water they can hold, and that presents a problem for them. If a plant's cells hold a lot of water, that means all of the things inside of a cell, like glucose, amino acids, proteins, and everything else, is more dilute. The cell, however, likes to maintain all these components at a constant concentration. To overcome this problem, plant cells have a large central vacuole to maintain osmotic balance. When excess water comes into the cell, the extra water is stored in the large central vacuole, thus maintaining a constant concentration of all those other cellular components. Now that we've seen how cells behave in different aqueous solutions, let's see why they behave that way. The selective permeability of membranes starts with a phospholipid bilayer. Recall that phospholipids have a hydrophobic tail composed of two fatty acid chains and a phosphate-containing head group that is hydrophilic, all linked to a glycerol backbone. Phospholipids in water will spontaneously form a phospholipid bilayer. Spontaneous means that it happens all by itself. You don't have to add energy or anything else. Phospholipids self-organize in a tail-to-tail configuration, making a hydrophobic core of fatty acid tails and a hydrophilic exterior of head groups, which are exposed to the watery environment either inside or outside the cell. This arrangement is called a phospholipid bilayer, which usually gets shortened to simply lipid bilayer, or just bilayer. Keep in mind that cells are three-dimensional, so the plasma membrane that surrounds the cell is like the surface of a sphere. Its key facet is that hydrophobic core, which is impermeable to hydrophilic molecules like glucose and amino acids, or ions like sodium and chloride. Biological membranes, however, are more than just phospholipids. They also contain protein. The fluid mosaic model describes biological membranes as a lipid bilayer with embedded proteins. For the sake of simplicity, let's just say that a typical biological membrane is about 50% protein and 50% lipid. The embedded proteins have many different functions. We will concentrate on transport proteins, but the proteins of membranes can also be enzymes, signaling molecules, structural proteins, and others. It's called fluid mosaic because the components are free to move laterally, that is, from side to side, but they can't flip-flop, that is, to move from the outer leaflet to the inner leaflet. As I said, we will concentrate on the transport function of biological membranes, and we will mainly study three different ways that molecules move into and out of cells: simple diffusion, facilitated diffusion, and active transport. We will also look briefly at exocytosis and endocytosis. Let me give each one a brief description here, and then I'll give an example of each. Simple diffusion and facilitated diffusion are both forms of passive transport. That is, they don't require the input of energy because molecules are moving down their concentration gradient from high to low. Simple diffusion is just what the name implies. Molecules simply pass through the membrane. Only molecules that can get through the hydrophobic core of the lipid bilayer can freely diffuse through membranes. Such molecules are hydrophobic themselves. In other words, they don't dissolve in water. Since the molecules can get through the lipid bilayer, they don't need a protein transporter. A consequence of the fact that simple diffusion is passive is that the direction of movement is dependent on concentration gradient. Molecules move down their concentration gradient. Facilitated diffusion, sometimes called facilitated transport, is like simple diffusion in that it is concentration dependent. Unlike simple diffusion, however, facilitated diffusion requires a membrane-embedded protein to transport a hydrophilic molecule through the membrane. In other words, the diffusion of a hydrophilic molecule is facilitated by a protein transporter. Active transport is like facilitated diffusion in that it requires a protein transporter. Unlike facilitated diffusion, however, it uses energy to move molecules up or against their concentration gradient. That's what active means. It requires energy. The energy comes from ATP, either directly, as in primary active transport, or indirectly, as in secondary active transport. I won't say anything about exocytosis and endocytosis now, but I'll give an example a little later. Here is an example of simple diffusion, the same one we saw earlier. In this case, molecules freely diffuse through the plasma membrane without the assistance of a protein transporter. Molecules are moving from an area of higher concentration, outside the cell, to an area of lower concentration, inside the cell. Depending on the molecule, of course, the situation may be reversed with the net movement going from in to out. Examples of molecules that move this way are oxygen and carbon dioxide. As oxygen, O2, flows into the cell, it gets used up. So oxygen is always higher outside the cell than inside the cell and will generally flow in. Carbon dioxide is just the opposite. Carbon dioxide is produced by cells, so carbon dioxide concentration will always be higher inside the cell than outside, so carbon dioxide will flow out. Previously, you learned that steroids are lipids, and as such, they can also diffuse through membranes without the aid of a protein transporter. So steroids enter and exit cells by simple diffusion. Facilitated diffusion is like simple diffusion in that it's a passive process. It doesn't require energy because molecules are moving from an area of higher concentration to an area of lower concentration. So it is a form of diffusion. And remember, there's a natural tendency for that to happen. That is, no energy required. Unlike simple diffusion, however, facilitated diffusion requires a protein transporter because the molecules being transported is not membrane permeable. Glucose is a good example. Recall that glucose is a hydrophilic molecule. It interacts well with water but not with lipid and therefore will not pass through the lipid bilayer. So a protein transporter is required. In other words, the protein facilitates diffusion of glucose across the membrane. Those are the basics about facilitated diffusion, but there's one other thing in the figure that's worth pointing out. Facilitated diffusion, because it's protein mediated, is highly selective. Take a look at the picture and you'll see there are two different kinds of molecules outside the cell, represented by the small hexagons and the larger ovals. And they both exist in a concentration gradient, high outside the cell and low inside. The protein transporter, however, is specific and only allows the hexagons through. This kind of selectivity is finely tuned, and transporters can distinguish between very similar molecules. For example, transporters can distinguish glucose from galactose, which are both 6-carbon sugars with a very similar structure. Even more impressive is that transporters can distinguish sodium ion from potassium ion. We'll see this kind of selectivity again in our next video, which is about enzymes. As an example of active transport, we'll use the sodium-potassium pump. This diagram is a little confusing, so let me walk you through it. It shows active transport of two different ions, sodium and potassium. Note first that sodium, the orange hexagons, is high outside the cell and low inside the cell, and potassium is just the opposite, high inside the cell and low outside. At left, you have sodium being loaded into the protein transporter, which is open toward the inside of the cell. The transporter then changes shape so that it is now open toward the outside of the cell, and the sodium moves away and has been transported outside of the cell, against its concentration gradient. It takes energy to move a molecule against its concentration gradient, and that energy comes in at the second step with the protein's shape change, which uses ATP as its energy source. We'll learn all about ATP and how the cell uses it for energy in the next video. With the transporter open to the outside, potassium can now move into the channel. Another shape change and the transporter is once again open to the inside, and potassium can move into the cell against its concentration gradient. Residual energy from the ATP powers the second shape change. The result of the sodium-potassium pump's action is a chemical gradient, with potassium high on the inside and low on the outside, and sodium low on the inside and high on the outside. We've talked about passive transport and active transport. There's another kind of transport that I'm just going to touch on, bulk transport, which includes exocytosis and endocytosis. Exocytosis is for stuff exiting the cell. For example, a cell might secrete a protein. Insulin is a protein that some cells secrete. That's what's shown on the left. This is an insulin secreting cell. Insulin, which is a protein, is contained in these membrane enclosed compartments called vesicles. In exocytosis, the membrane of the vesicle fuses with the plasma membrane, and anything that was inside the vesicle is now outside the cell. So the cell has now secreted insulin. And for our purposes, we can say that exocytosis equals secretion. Endocytosis is bulk transport into the cell. Endocytosis is for stuff entering the cell. This picture shows endocytosis. I won't go into the details. You can study them in the picture for yourself. Let me just say that endocytosis is the way that certain nutrients get into the cell. Iron and cholesterol are examples. But endocytosis has many functions beyond uptake of nutrients. It is also the way that some viruses, like the coronavirus that causes COVID-19, get into cells. Okay, let's bring it back to water. Remember, molecules that are membrane permeable enter the cell by simple diffusion, and molecules that are membrane impermeable get into the cell via a protein transporter. Water is kind of in between. Water is polar and not membrane permeable, but it's small enough and abundant enough that it will to some extent cross membranes without the aid of a protein transporter. But it does so very slowly and in small quantities. When water moves quickly or in large quantities, it uses a protein transporter. In other words, by facilitated diffusion. And that's what this slide is showing. The protein transporter for water is called aquaporin, as in a pore for water. The existence of a protein transporter for water had long been predicted, but it wasn't actually discovered until 1992 and led eventually to the Nobel Prize for its discoverer, Peter Agre. The graph on the left shows that control cells, which do not have any aquaporin, remain stable in hypotonic solution like fresh water. If you measure their volume, it remains the same over several minutes because water is not coming into the cells. In other words, the control cells are impermeable to water. If, however, you put aquaporin into these cells, they swell over the course of 3 minutes because water is rushing in. This shows that when cells have aquaporin, they are permeable to water, but without aquaporin, they are impermeable to water. The pictures to the right show the same thing. Cells without aquaporin, here on the bottom, do not swell in fresh water. The cells on top have aquaporin, and they swell and burst after just 3 minutes. The upshot from the last slide is that cells differ in their permeability to water due to the amount of aquaporin they have. Red blood cells and most plant cells have lots of aquaporin and are very permeable to water. Frog eggs, the cells used in the last slide, have no aquaporin and are impermeable to water. They have to be. Frogs lay their eggs in fresh water. In still other cells, the permeability to water is regulated. An example is collecting duct cells of the kidney, and that's what's shown in the figure. The kidney makes urine, as you are probably aware, but the body doesn't want to lose too much water to urine. Part of the job of the kidney is to minimize water loss by reabsorbing water during urine production. How much water gets reabsorbed depends on your hydration state. Under normal conditions, water passes through the kidney with relatively little reabsorption. If you're dehydrated, the kidney will absorb more water. The kidney does this by putting more aquaporin at the surface of kidney collecting duct cells. The left part of the figure shows the normal situation. When you're fully hydrated and the kidney doesn't care about reabsorbing water, aquaporin, the orange ovals, is present in the cell, but it's not at the surface where it can affect water uptake. The right side shows what happens when you're dehydrated, leading to lower blood volume. The aquaporin that's present in the cells is moved to the cell surface where they can increase the reabsorption of water. The larger point here is that molecules whose transport is protein mediated are subject to regulation. That's true for water and other molecules like glucose, sodium, and calcium. In this video, we looked at the structure of biological membranes, which is described as a fluid mosaic. This structure leads to the selective permeability of membranes. How a molecule moves across a membrane, simple or facilitated diffusion, or active transport, depends on the nature of the molecule and whether it moves with or against its concentration gradient. In the next video, we'll learn the energetics that govern these and other biological processes, and we'll learn about the enzymes that control chemical reactions.